Hello, my name is Pavel. I'm a first-year graduate student here in Stanford Burnham Prebys, working in Francesca Marassi's lab. And today I'd like to give you a little bit about information about my work. And the title of this presentation is Nuclear Launch Strike Against Cancer. And I'm going to be using this metaphor throughout my talk, but don't be alarmed. There is actually no nuclear launch strike involved. And to really put it into perspective, when we talk about cancer, I got to give a certain perspective in terms of what do we talk about. Now, initially, when we think cancer, we know it's a devastating disease that affects a lot of people around the world. However, what we really talk about is that it affects their tissues and organs. However, we know that the tissues and organs are comprised of cells, which in turn are regulated very heavily by all the different molecules inside of them. And I want to keep that in mind, because what I'm going to be talking about are the molecules that are regulating this cancer process. Now, when you watch a movie, for a second, let's go a bit away from the cancer. When you watch a movie, you remember this moment when there are two guys sitting in a room turning the keys to launch a nuclear missile. If you can remember this moment, we know that that's actually not real. That's not how it really happens. Primarily because no one's going to tell us how it really happens, right? This is all classified information. And however, what we do know is in real life, we have cells that are capable of doing exactly the same thing. They're capable of doing self-destruction by essentially having those kind of kill switches that need to be inserted somewhere that in turn just destroy the cell. Now, what we really want to achieve here, especially what I'm trying to achieve, is to declassify this information and to present it in this fundamental knowledge base to really make sure that we are able to program the cells to self-destruct. And we want to do that by doing, for example, drug discovery. And essentially, if we have those cells that are able to self-destruct, we can essentially destroy the cancer and prevent it from happening, for example. So how much do we already know about it, and what are some of the questions that are pertaining to those nuclear launch keys? Now, what are really the questions that need to be answered are: what is the shape of all those things? Where are they stored? What are those guys wearing, maybe? But okay, maybe not that. But what are the key? How many keys do you need, and what safeguards are in place to control this such complicated mechanism? And what I want to draw attention to is that all those questions have important analogies in cancer biology. And just to name a few, those molecular keys essentially could represent molecules that need to be inserted in some place within the cell. And there is a specific mechanism that governs this particular conformational change, if you will. And there are a whole other bunch of other molecules that regulate this process. Now, to introduce the molecule that I'm investigating, this is that molecular key that is essentially a kill switch for the self-destruction of those cells. On the left, you could see the Soviet nuclear launch key. I don't know if it looks exactly like this, but bear with me on this. And on the right, the protein that I'm investigating. I admit they don't look very much alike, but what is important is that in nature, those proteins that are called PACs, they are much more flexible and are under very tighter security, unlike the keys that are used for actual nuclear launches. And what essentially happens is you have this inactive protein on the left, which actually changes its structure in such a way that it could be inserted in a particular place within the cell that in turn activates this self-destruct mechanism. And this is in fact what we're trying to investigate. Because if we're able to specifically manipulate this process and know everything about structure and all there is to know about the regulation and safety of that process, we may be able to control it and program the cells in which this process occurs and maybe treat cancer in this way. So this brings me to what my project is really all about. It is to determine the shape of those molecular nuclear launch keys, and understand the mechanism of their action. And the way I'm using the technology is I'm using the imaging process that is very similar to brain scanning with like MRI, but on a much smaller scale. So essentially I'm using the same principle to study the shape and the changes in that shape of those molecular keys. And the application of that would be to study the drug discovery for specific programming of those cells to self-destruct at a given cancer. So with this, I'd like to give a huge thank you to my lab, which have been helpful in me pursuing this project. And I just wish that it would not be too late for many people who needs this kind of understanding and declassifying of this information. Thank you very much.